You hear the expression, take a sample and check it. Is it so easy just to take a sample? What do you need to know before you take that sample in order to rely on the answer? How are you sampling? Is your results worth having? Well, let's take a look at some questions before we start answering that question. Is the flow constant? Is the concentration of analyzed substances constant? If not constant, how does flow and concentration vary? Answering those questions, it will also guide you to the correct sampling methods. What sampling method do we have? Manual sampling, basically a grab sample. It only shows you the concentration at that specific point in time when you took the sample. This you can use if you know what the concentration should be when you sample. You have a fixed value. So basically it's a checkup. If it's off, then you have to do some other sampling and maybe correct the process. Now, automatic sampler gives you representative values, and they can use time between sampling, and they can also sample based on flow. And here on the right hand side, lower right hand side, you have a picture of automatic sampler. In this case, quite advanced. I think at this time I used 24 bottles to sample stormwater, I think. Now we look at the sample vessels, collecting a sample and then off to the lab. Minimum volume for an analysis. Depending on if you're doing metals or some organic substances, the volume can change dramatically. This you always need to check, and the best way, check with the lab. Type of bottle you need. When you take a sample, depending on the substance, again, you use different types of bottles. On this picture here, you see plastic bottles, a small one, a bigger one, I think a liter one, and then you have a green flask for an organic substance that I was testing at the time. So this is also important to know, and again, check with the lab. Here comes an important factor. Contamination risks depending on substances. Some substances are in so low concentration, and also these substances are not only in the water but could be in the clothes you have, in the air. So when you sample certain specific substances, you need to scrutinize every step of the sampling procedure. Basically how you order the flasks, what type of flasks, so the flask doesn't contaminate the sample. How you sample, so you don't have clothes that could emit some of those substances. Most of samples or parameters are not difficult. Standard thing, well flushed sampling pipes before collecting sample volume. This is a picture from a sampling we did where we took a sampler from another place and installed it at an intake point. And in this case, we had very long tubes going down. And one of the things we were very careful with was flushing the tubes. Sometimes you have to change hoses to a sampler after each sample. Some very specific times, but very uncommon, but it could be. But basically, standard equipment, automatic samplers always check the flush times. Also check the hoses going down to the sample, so they haven't been growing in them or contaminated some way. I remember one time in a drinking water plant, we took some special samples, and suddenly we had bacteria popping up in our tests, and we couldn't understand why until I realized that the hose we had going into the sampler was very long. So over time, bacteria had started growing in this hose. Next thing, marking of the samples. Know them when you read them. Over the years, I've been in all kinds of water treatment plants, sewage treatment plants, process plants, you name it. And one of the things when I've been working there, sampling is a small part of the job, but there is a lot of samples going on. And when you mark a sample, be kind to yourself and have longer text. In this case, you see here in the picture, I have the date, place, and also what unit I was sampling. So when you get the results back from the lab, you don't need to go into a list to check what sample it is. Because many times you make it easy to yourself. You put 1A, 1B, 1C. And sometimes it could be sufficient, but be a little bit, take some more time marking the samples. Now, next, keeping sample cooled. Not so much to say about that, quite standard. Also, transport to the lab. When you've taken the sample, try to get it to the lab as soon as possible. And always check with the lab, are there some analysis you take there that you can't store more than maybe 10, 12, 24, 48 hours. Always check this. And also, to what lab is the sample transported to? Today, you can take a sample in one country and the lab will send it to another country for analysis. This could have some impact on the results as well, but you need to be aware of this when you check the results that you receive from the lab. When you get results back, always scroll through them, even if you have many other things to do. Take some time when you get results to check them. And here again, when you mark the sample good, it's easy to see if there is some results that is way off the scale. In this case, you immediately call the lab back. Because today, if you leave the results lying and it goes too much time and there is some error, sometimes they can't do the analysis again. 2 pictures. If you come to the island of Gotland, I think you will recognize these environments. That's all I'm gonna say, except for thank you, and remember, water really, really rocks. Take good care. Talk to you soon. Bye bye.